From the CII. It's going to talk about some of his research. Next week we have actually Claudia Angeli, so she's doing spinal cord injury. She's to come next week as well. Dr. Boyd actually hails from Texas, so he's like me, a little bit ways from home, but he's been here since 2008. Is that
yes.
right? 2008.
End of 2008.
His postdoctoral fellowship was at the University of Georgia, and then he did his PhD at Georgia Institute of Technology. So he's going to talk a little bit about hypercholesterolemia. I can't say it. Maybe he's better at it than I am. So.
Okay.
Dr. Boyd.
Thank you. Okay, so this is an unusual setting. So if I look a little odd a little oddly, that's just because I'm not quite accustomed to seeing everybody spread out quite like this. Thank you for the opportunity to come over here. Guru had asked me a couple times, and I procrastinated, and he finally asked me again. And I thought, I really should do this. So, I was going to. So first of all, if you have any questions, I know you're going to be looking at some of the different screens. Make sure you wave your hand or something like that at me so I get your attention. Or if I don't see and you have a question, just holler out. So I was going to present the talk in 4 sections. I was going to just give a little bit of background of my career very briefly. And then a little bit of background on cardiovascular disease and cholesterol, the LDL receptor, and I'll define that, and familial hypercholesterolemia. And then we'll talk a little bit about my research. And again, if you have any questions, just raise your hand, call out, whatever. All right. So my background. So I'm a native of Dallas, Texas. Graduated from high school in 1976, so you engineers can calculate my age very quickly. You should be able to. When I graduated from high school, I went up to the University of Arkansas and majored in agronomy initially. I thought I was going to be a farmer. After 2 years, I dropped out for a year and worked in my family's business. My family had a wholesale company that sold truck parts, wheel attachment parts, brakes, and suspension components. After a year, I matriculated into a small school called Ambassador College in Pasadena, California. And then I graduated with the Bachelor of Arts in 1982. Went back to work. I actually got married. My wife is back there. We've been married 37 years. I went back to work for my family business and held a number of different positions, from entry-level purchasing, and then I went back and I got my mechanical engineering undergraduate from the University of Texas at Arlington in 1990. So I was responsible for purchasing, engineering, quality control, and a number of other things. I started getting interested in biomedical engineering partly through when I started volunteering at Baylor Hospital in downtown Dallas in the emergency department. I did that, I think, about 3 years, twice a month. We had the family decided we wanted to sell the business, so I was kind of a unique opportunity to change careers. And in 1996, decided that I was going to go back and get my master's degree in bioengineering. And again, that was partly due to my experience volunteering at the hospital. I did my research, my master's research, at the Orthopedic Biomechanics Lab at the University of Texas Medical Branch in Galveston, which is an island along the coast south of Houston. And then I initially actually started my PhD at the University of Alabama, Birmingham, in the engineering department. But my first mentor ran out of funding. So he didn't lose his funding per se. It had to, he ran out of funding, and so I actually found my second mentor while training for the local marathon there in Birmingham, and we ended up running the marathon together. And so I kind of had my interview with him doing that. And so my first question was, would he take me after the marathon? A couple of days later, I went to talk to him. My first question was, would he take me? And he said yes. My second question was, are you planning on moving? And it turns out that he was investigating, looking to move from UAB, and he took a position at Georgia Tech Emory. So I transferred from UAB to Georgia Tech. The lab was in the Emory Cardiology, and then I finished. I graduated 2003, and then I started my postdoc in 2004 at the University of Georgia. At the time, this was really kind of the beginning of the expansion of human embryonic stem cell research. And UGA, actually, the lab that I worked in was in the Animal Dairy Science Department. And it was there because the PI that I worked for, his background was in somatic cell nuclear transfer and in animal, regeneration, and then he was really his work was evolving, and he was then working on human embryonic stem cell work. So I focused on trying to differentiate HESC to endothelial cells aligned with blood vessel, and also different mesenchymal lineages. So I was hired in 2008, late 2008, as an assistant professor, and my lab was in the CII. Initially I was in the Department of Surgery, and then transferred to the Department of Physiology, and I'm currently associate professor. My lab's still in the CII. So, cardiovascular disease. Okay, so I've already given the hint, but basically what does this represent? Anybody?
Fried chicken.
Say it again?
Fried chicken.
Fried chicken, yeah, there you go. Okay, so this is a map of cardiovascular disease, and you can see that Kentucky here is located kind of in the middle of cardiovascular disease zone. One of the highest incidence occurs here in Kentucky. And you probably are familiar with the risk factors. So high blood pressure, high cholesterol, diabetes, smoking, inactivity, poor diet. So what we're going to focus on today is the role of high cholesterol, and then we'll get into the genetics of hypercholesterolemia and what that causes. So probably most of us know LDL cholesterol, or low-density lipoprotein cholesterol, is frequently called bad cholesterol because when you have too much of it, it leads to the development of atherosclerosis, which here. So you can see that when you have high cholesterol, one of the things that happens is you begin to get lipid deposit into your arteries, and interestingly, you don't develop cardiovascular disease, atherosclerosis, in your veins. And can anyone have any idea why that occurs? Any ideas? Let me give you a hint. It also occurs in locations where you have branch points. Okay, so like like in the aortic arch, you have a curve, this big curve in the aorta, and you develop atherosclerosis on that inside of the arch, inside the curve.
Shear stress?
Differential shear stress, you get separation of the laminar blood flow, and so at branch points, those are places where you tend to get disturbed flow, and so if you think of like a bend in a river, where would you expect to see like the sandbar deposit? It would be on the inside of the river where it's flowing slower, and that's basically how it happens in the arterial system. So going into the LDL receptor and FH. So the importance of cholesterol in biology, I think, is it's interestingly represented here in that 15 Nobel Prizes have been awarded to folks who have done their research on various aspects of cholesterol metabolism. So how is it regulated? How is LDL cholesterol regulated? So primarily it occurs in the liver. And the parenchymal cell, or the functional cell of the liver, is called a hepatocyte. Okay? So this glove and stem here I'm representing as the low-density lipoprotein receptor. And on the LDL receptor is expressed on the hepatocyte, and of course it can be expressed on other cells within the body as well. But the liver is the primary mediator of LDL homeostasis. And so you get the LDL receptor, or the LDL particle binds to the LDL receptor, and it's internalized and then metabolized. So when you have high cholesterol, and you have all these different LDL particles, your physician is going to, the first step will be that they will recommend that you start taking a statin. And statins block cholesterol synthesis. Does anyone know what that rate-limiting step is that the statins block, or statins inhibit? Anyone know the enzyme? HMG CoA reductase. So anyway, statins block the rate-limiting step in cholesterol synthesis. And then what that causes, statins don't directly upregulate the LDL receptor gene expression. They actually block HMG CoA reductase, and then that causes a downstream reaction that then upregulates expression or transcription and then expression of the LDL receptor. So statins essentially block endogenous cholesterol synthesis, okay, so it stops your hepatocytes from producing cholesterol, and then it also forces an upregulation in the LDL receptor, which then gives you a greater ability to internalize cholesterol, the circulating cholesterol, and control how much cholesterol you have in your body. Now, some folks, so that's the way it works if you have a normal or wild type LDL receptor. But some folks have genetic mutations in the LDL receptor, which cause the LDL receptor to become dysfunctional. And that is termed familial hypercholesterolemia, and it's familial because basically it's an inheritable disease passed on from both parents, or it can be passed on from both parents, and it leads to hypercholesterolemia, or high cholesterol. Okay, so the LDL receptor gene is located on chromosome 19. If you have one allele that is affected, one of the genes, one of the pair that's affected, that is referred to as heterozygous hypercholesterolemia, okay, or familial hypercholesterolemia. If you have both alleles, then it's generally referred to as familial hypercholesterolemia. So, but there's another condition where you could have both genes or both alleles affected, and that's called compound heterozygous familial hypercholesterolemia, or FH. What would a compound heterozygous be? What would its genetic condition be? So if a heterozygote has one mutation, and a homozygous has the same mutation on both alleles, what would a compound heterozygote be?
Different mutations on each.
Yes. So a compound heterozygote has inherited different mutations on the alleles. For simplicity, it's often just referred to as homozygous because it ends up clinically having the same effect. But these are actually two different circumstances. Okay, so what are the effects of homozygous FH? So for a wild type, person with wild type LDL receptor, if you have LDL cholesterol, this is not total cholesterol, but LDL cholesterol greater than 190 milligrams per deciliter, okay, which is like right here, then again, that's the time that your primary care physician is going to start prescribing or suggest that you start taking statins. Okay, so a heterozygote, okay, heterozygote FH is going to have LDL cholesterol in the 250 to 500 milligram per deciliter range. So, more than 2 times what a normal person would have. And then a homozygote is going to have a range of 500 and approach up to 1,000 milligrams per deciliter. So they can be close to 5 times what a normal person would have that would be considered high cholesterol. Although homozygous FH is considered a rare disease, so there's not that many people that actually have it, but in the literature, it's not that uncommon that children under 10 years old are reported to have clinical symptoms of cardiovascular disease. And this is one of the extreme examples. This was reported in 2011. So a 4-year-old who was asymptomatic had just dropped dead suddenly, and ended up that they had a 98%, their coronary artery was 98% occluded, and this is a cross-section of their coronary artery. It turned out that they had LDL cholesterol in the 800 milligram per deciliter range. So for these patients, it is an extremely, even though there's not that many, these patients have a lot of problems, and they typically don't live much beyond their second decade. So what are treatments for these people? This ezetimibe it blocks cholesterol absorption in the intestine. PCSK9 inhibitor, PCSK9 actually binds to the LDL receptor and causes it to be prematurely degraded. Inhibitors PCSK9 are also used to extend the life of the LDL receptor. But the caveat for PCSK9 inhibitors as well as statins are that you have to have functional LDL receptors. If you, as a patient, don't have a functional LDL receptor, then neither one of these treatments is going to benefit you. Lomidopide is a microsomal triglyceride transfer protein inhibitor. Basically, it blocks the transfer of triglycerides into the LDL particle. And so it essentially inhibits the production of LDL cholesterol being produced in the cell. And then lipid apheresis is kind of like, it's a mechanical removal of lipids, and it functions kind of like dialysis. But it's very expensive. For these patients, if you take this and lomidopide treatment together, just those can cost over $500,000 a year. So it's very expensive to treat these patients. And then, of course, liver transplant is also an option at the for the most extreme patients. In addition, it's not uncommon that when someone's going to have a liver transplant, that they've reached the point that they also have to have a heart transplant. Okay, so people have been, because FH is caused by a single gene mutation or a single gene dysfunction, it has for decades been the target of gene therapy. More recently, people are starting to look to see if cell-based approaches could also be used. And then combining these into both because we have the stem cells that can be differentiated into hepatocytes, combining these approaches to see if you can make both a gene and cell therapy approach. So what is my lab working on? One of the things, we have been generating induced pluripotent stem cells, and I'll describe that in just a second, that are derived from FH patients. We have been utilizing a couple of different approaches for introducing functional LDL receptors into these cells. We have been taking these cells and deriving them into hepatocytes. If you remember, we would want to do that because the hepatocyte is the primary liver cell, or the primary cell in the body that controls cholesterol homeostasis. And then we've been combining these with other cell sources, stromal and vascular support cells, to generate liver tissue organoids. Okay, so first of all, what is a pluripotent stem cell? So a pluripotent cell can change and differentiate into all the cells that make up the body. So in development, the cell that, the most potent cell, which is called totipotent, is the zygote. So what's a zygote? Anybody know what a zygote is? When do you have a zygote?
Immediately subsequent to fertilization.
Exactly, that's right. Thank you. Thank you. So a zygote is a fertilized egg, right? And it is a single cell, and a zygote is referred to as being totipotent. And what that means is that it can develop into all of the cells, not only that form the body, but also the extra embryonic tissues like the placenta. Okay? So a pluripotent cell, and human embryonic stem cells, iPS cells, are considered to be pluripotent cells and not totipotent, because they can generate all of the cells of the body. Now, this has been proven not in humans, but in animal models. Okay. So there are three sources of pluripotent cells. So the first, as I mentioned, are somatic cell nuclear transfer, and that's where you take an egg and you enucleate it. So you take the nucleus out of the egg, and then you transfer the nucleus from a terminally differentiated somatic cell, meaning like a fibroblast, or an endothelial cell, or a mesenchymal stem cell. Okay, and you take that nucleus and you insert it into the enucleated egg, and that starts to differentiate and form. Well, it will actually go all the way to form an organism, but you can isolate pluripotent stem cells at the blastocyst stage. Human embryonic stem cells are another source of pluripotent cells, and then induced pluripotent stem cells, and we'll go into that in just a second. So these are actually some pluripotent stem cells, iPS cells, that I actually have growing. I took this just the other day in the lab. We also introduced a GFP transgene into the cells so they fluoresce green. So some advantages to using iPS cells. First of all, they can be made patient-specific, meaning you can take a cell from a patient, as an example here, a patient with FH, and you can make iPS cells from them. Now you can do the same thing with somatic cell nuclear transfer, but to do that, you have to also have an egg donor, okay, which is not trivial. The other advantage is that there's no embryo destruction, which is a major bioethical concern. With both SCNT and human embryonic stem cells, to get those cells, you have to destroy an embryo. You don't have to do that to generate iPS cells, okay? So how do you make iPS cells, okay? So this is kind of a graphic for normal development. So you start up here as the zygote, the totipotent cell, and with time and development, you get greater and greater specification, and you lose, your cells become more terminally differentiated and specified, and they lose potence. So if you start here, it's a totipotent cell. This might represent a mesenchymal stem cell or a hematopoietic stem cell, and then as you go down down the hill and through these different valleys, they become more and more specified. So zygote, more specified. Examples would be bone, brain, or liver. Okay, so again, you take to generate iPS cells, you take a terminally differentiated cell like a fibroblast, a peripheral blood mononuclear cell. So those iPS cells that I showed you, those were actually derived from peripheral blood mononuclear cells that we took from a donor. You can take the epithelial, you can isolate the epithelial cells out of urine. You can take keratinocytes off of a hair follicle, and there's other cells that have been used. Fibroblast and PBMC are probably the most common, and certainly fibroblasts have historically been the most commonly used. You take these cells and you introduce these 4 transcription factors that are commonly referred to as the Yamanaka factors after the scientist Shinya Yamanaka, who discovered these and was actually awarded the Nobel Prize in 2012. So you introduce these 4 transcription factors, and these are expressed at the very earliest stages of development. And so when you introduce these 4 transcription factors into a terminally differentiated cell, it essentially causes them to go back up this slope from the terminally differentiated state all the way back to the pluripotent state, all right? We've actually generated iPS cells from 9 different patient sources. This is a list, we've submitted this for publication. This is a cohort of FH-IPS cells that we have generated from patient blood that was donated. So we have a collaboration with a physician in the Rogosin Institute up in New York City, and she consented her patient. So why is this important that we consented the patient to get their blood to do this? Why does it matter that I told you that we consented these patients? Can anyone think of an example, a historical, very famous example of someone who wasn't consented and their cells have been used even today? You may be culturing some. Henrietta Lacks, that's right. So we consented, we got their consent for them to donate the blood, and we told them what we're going to use it for. The blood was shipped down UPS next day air. We isolated the peripheral blood mononuclear cells and then generated the IPS cells. We reprogrammed them using vectors that are in a vector that's called a Sendai virus. So it's a transient transduction. Okay, so the vectors don't stay around. It turns out that of these 7 patients, we had 4 that were homozygous. Okay, so if you remember, that's having the same mutation in both alleles, and 3 are compound heterozygotes, so different mutations in each allele. Our naming nomenclature, L stands for Louisville, R for Rogosin, and these are actually numbered in the alphabetical order of the patient's initials. We have 2 that are female and the rest are male. You can see here at the bottom, LR7, their LDL cholesterol when they were initially started was 550. And it ranges from this all the way up to 907. So all of these are extremely high. And so we have number 2 and number 5 that actually have the same homozygous mutation. They're not related in any way, but all of the others are different. So it's one thing that's really important. So this is the LDL receptor gene, and this is what the protein looks like. There are over 1,700 identified mutations that occur in the LDL receptor. And certainly not all mutations affect the receptor function equally. Some are much more devastating than others. Some completely block transcription, and some just delay or reduce the efficiency of the LDL receptor. There are 6 different classifications depending on where the mutation affects processing of the LDL receptor. So a class 1 occurs where it actually completely blocks RNA synthesis. Class 2 is broken up into an A, B. So it's called a transport mutation, but actually what happens is the mutation causes misfolding of the protein. And so if it's class 2A, the misfolding is so extreme that it gets stuck in the ER, in the endoplasmic reticulum. If it's class 2B, the misfolding is not so extreme to where the protein can escape, But even though it may reach the plasma membrane, it still may not really be able to bind the LDL cholesterol. A class 3 affects the binding of LDL to the LDL receptor. Now there's some other non-LDL receptor mutations within the genome that are referred to as familial hypercholesterolemia, but we're not going to go into those. A class 4 prevents the LDL receptor from aggregating in the clathrin-coated pit. Okay, and if you remember from your biology, clathrin-coated pits are the basic, are the fundamental mechanism that regulates endocytosis, right? And so actually, the LDL receptor and FH was the model that was used for determining receptor-mediated endocytosis. And FH was that model, okay? So a class 5 mutation, the LDL receptor can aggregate in the clathrin-coated pit, and it can be internalized in the endosome, but it can't separate and then be recycled. So normally, it comes in, the LDL particle separates from the receptor, and then the receptor is recycled up to the plasma membrane, binds again, goes to the clathrin-coated pit, and recycled, okay? But in a class 5, it is, the receptor can't separate from the particle, and so it's prematurely degraded, all right? There's a class 6, which has a mutation in the cytoplasmic tail portion, and it fails to actually reach the plasma membrane. Okay, so this is a Western blot showing the protein expression of those 7 patients that we have made iPS cells from. These are all treated with statins. BL3 is actually from the cells that I showed you previously. This is a normal LDL receptor expression, and you can see that there are 2 bands. Okay, so this bottom band is what's called the immature state, and it's 120 kilodaltons. Okay, and it shows up when the LDL receptor gets stuck in the ER. All right. The mature band is when the LDL receptor then moves out of the ER to the Golgi, it's modified, and then goes up to the plasma membrane and can function. And so you can see here, so this is just a recombinant protein, LDL receptor protein, used as a positive control for the antibody, for the Western blot. So you can see here on this wild type cell that all of the LDL receptor protein is showing up in the mature form, all right? But here, for LR1 and LR4, and actually LR3 as well, there are both forms, okay? So there's an immature and a mature. And so what this suggests is that all 3 of these are class 2B mutants, okay? Because there's some of the immature and there's some of the mature showing up. We don't know how functional these mature proteins are, but the fact that there's both immature and mature suggests that there's protein that's getting stuck in the ER as well as some that's escaping, all right? Now here, 2 and LR5, there's no protein showing up, so this could be a class 1. LR6 and 7, there's very low levels of only mature that's showing up. So these could potentially be a class 4, 5, or 6, all right? Okay, so, all right, so we published this in 2017. So GMO 3040 is from a patient that well, we got the fibroblast cells from the Cohort cell repository. It was a male, 9 years old, and at the time it was tested, had LDL cholesterol of 820 milligrams per deciliter. So again, extremely high. You can see here, so these are IMR90, our fibroblast line that have a wild-type LDL receptor. This is these are the fibroblasts for the GMO 3040. So lovastatin, when you add statins to it, it upregulates the LDL receptor, and when you treat it with a fluorescently labeled LDL, okay, then if it's a regular wild-type LDL receptor, you can see it fluoresces very brightly. Okay, but here, there's basically no fluorescence at all, right? Now, the reason I'm showing you the excess sterols, if you add in excess sterols to your culture, it actually can cross the plasma membrane without needing an LDL receptor, and it downregulates the LDL receptor, okay? So you can see here, even in the wild type, when you add excess sterols in, it downregulates expression, and it doesn't take in any LDL receptors. So this is just showing that you have normal feedback function of the LDL receptor. We did a Sanger sequencing to find out what the mutation is. And so this is the wild type showing normal, what the normal gene would look like. Here it is showing that there is a missing 3 base pairs in this patient's cell, and that it is homozygous. Okay, so show this. And anyway, this is what the specific mutation is called. It is referred to as, in the literature, as either FH Lithuania or FH Piscataway. So FH Lithuania, because this particular mutation has been found in Lithuanian Jews who have also migrated in the late 19th century to South Africa. These are the fibroblasts. This is the iPS cells that we reprogrammed. We introduced the GFP vector into it. They can be differentiated into hepatocytes. So what we wanted to do was to use CRISPR-Cas9 to repair the 3 missing base pairs. And so we used a Cas9 nickase, and the nickase form of Cas9 actually only, so the regular Cas9 will cleave both strands of the genomic DNA. The Cas9 nickase only cuts one side. And the reason that we do that is, or wanted to use that, is because it is a more precise, it utilizes a more precise homology or precise repair mechanism called homology-directed repair. But if you use this, you have to have 2 guides, repair guides, that bind to your DNA sequence of interest. Okay? And so these are the sequences that we used. We also, this is the repair template, okay? And so right here are the 3 missing base pairs. This template would insert into right here, okay? And so this is the proto-associated sequence motif, and it has the Cas9 that we used uses the TGG PAM sequence for recognition. We also introduced a novel restriction enzyme, this XMN1. So essentially, and then we also introduced silent mutations here where it's red. Okay, so we made these changes. And even for this XMN1, we also introduced these silent mutations. So why would we want to introduce silent mutations that in particular associate with the PAM sequence here and then upstream from the PAM sequence over here? Anyone have an idea? Any reason why we would do that? So Cas9 requires a PAM sequence for the target sequence to bind to and for the enzyme to be able to recognize where it's going to be cutting. If we introduced these silent mutations in here to where it disrupted the PAM sequence, what would that do after you get a repair? Any ideas? Any guesses? If it requires a PAM sequence, but you change it after you've made the repair, what would that do? Would the PAM sequence be able to bind?
It doesn't duplicate.
It wouldn't know where to go. So basically what happens, the reason we did this is because if we actually got repair to occur, okay, we only wanted it to happen once, right? And so by changing the PAM sequence here, this target can't bind it again, all right? Now, the problem over here, we put these silent mutations downstream of the PAM sequence. We couldn't modify this and still get, if we would have modified this actual sequence, it would have changed the protein, the codon for the protein that's being coded here, so we couldn't change that. So we changed the upstream sequences up here to prevent binding. Now the other reason that we introduced this novel restriction site, I'll show you that in just a second. Okay, so this was our protocol. So we introduced by electroporation. We sorted for double positive, so that if we introduced these GFP and both of these guide plasmids that were RFP positive, if they were double positive, that meant that everything got in there. We then sorted for GFP and RP, and then we sorted for expression of the LDL receptor. We clonally sorted on and cultured them, and then we did our analysis. Okay, so we used what's called restriction analysis for looking at the product size on these cells, all right? So the PCR product for our sequence was 576 base pairs. If the XMN1 cuts within that sequence, then you will get a product of 384 base pairs and 192 base pairs, all right? So this is the untransfected or unrepaired cell line. If you add in the enzyme, it doesn't cut, right? Whereas for this clonally isolated cell line, here, if you add in the enzyme, you get the two. It cuts. The enzyme cuts. And you get these two products, indicating that you actually have a homozygous repair. If you had these three showing up, what would that be? So if you have a homozygous repair, you only get these two bands. If you don't get any cut at all, then you get this one band. If you had a heterozygous repair, what would you get? You get three bands, right, because one allele is not cut while the other one is. So basically what this shows, again, this is the unrepaired cell line. You get this is a class 2, so you get the immature and a little bit of mature protein. These are the repaired cells, all of the LDL receptors showing up in the mature form, suggesting that we had a complete and total repair of these cells. So We then took these cells and we were looking to build an organoid with them, but we needed other cells to support the stromal and vascular support of these cells. So we take adipose tissue, this is fat from liposuction, and you can isolate the fat and digest it. This is adipose cells. In the bottom here, you really can't see it here, but in the bottom are all the vascular cells, stromal cells, mesenchymal cells, fibroblasts. And if you put these in vitro culture, this is what it looks like in their phase, and they will start to form these in vitro well, these aren't blood vessels, but endothelial networks. If you implant them in vivo, this is actually from a GFP rat, and has been implanted into a non-GFP host. So, and then the red is, over here, is a dextran rhodomine. So anything that's red is a perfused vessel. So where it is red only is the host vasculature. Where it is green, this is from the donor GFP rat. And then where it is green and red is perfused vasculature from the donor. This is where anastomosis. So you get the host vasculature and the anastomosis from what's called the stromal vascular fraction. Over here, this is from the donor fat, and these are blood vessels from that we've implanted. And these are hepatocytes, and you can see that the blood vessels are forming a cage and supporting the implantation of these hepatocytes. So if we take our derived hepatocytes and we combine it with this stromal and vascular support, we can form tissue organoids, liver organoids. This is from phase, and then the green here are the hepatocytes. Now, one of the challenges that we encountered is, even though we can form these vessels by themselves, when we combine them into these structures, we don't know exactly why, but we have not been able to actually get a robust vascularization along with the hepatocytes. We can have stromal cell survival, but we are not getting this robust vascularization. So we could use these organoids for liver toxicity. We could potentially use them as a liver developmental model. We could use them for cholesterol metabolism, and you potentially at some point could use them as some kind of a modular assembly to potentially even build up a liver tissue or a liver organ. So to summarize, we know that cardiovascular disease is the #1 cause of death in not only the U.S. but in the world. Elevated cholesterol is one of the primary cardiovascular disease risk factors. There are genetic mutations that can exacerbate cholesterol deregulation, and then combining cellular and genetic technologies may allow us to be able to develop alternative therapies for these kinds of patients that have FH. So if you have any questions, I'll end it here. I think we have a little bit of time. Any questions? Any questions? Yes?
So have you tried re-implanting these into like a mouse model or something?
So we've done some implantation directly into the liver. We have not really gotten much survival. That's really why I didn't show that work. That's one of the big issues with trying to implant something like this is you really have to get anastomosis very quickly, else your cells don't survive, and so we're still working on how do you get this the vasculature to form within the organoid sufficiently so that when you implant it, it actually hooks up, and then you get a vasculature that supports engraftment. Does that make sense?
What if you just put the hepatocytes in a liver? Would they?
So you'd still have the same, you'd still have the same issue is, I mean, there are a lot of people in trying to, not just for FH, but for liver disease, a number of liver diseases. One way to approach it is to just take your hepatocytes and try to implant them directly. And so one of the primary ways of doing that is injecting them into the liver through the spleen, but the cells have to get out of the sinusoids and and then engraft, right? So that's one barrier, and even when you temporarily disrupt the endothelial sinusoid barrier, again, they still have to get in and engraft into the host tissue, and that's a major problem. Most of the hepatocytes that are transplanted that way don't survive, and it hasn't been overcome yet. Yes.
I was just going to ask, on those organoids, do you have a construct or a scaffold that they're grown in? And have you considered to get the vasculogenesis signals, like the angiogenesis signals, incorporating those in to help form this? So we based what we were doing. So there was a paper published by a group. The investigator's last name is Takebe. And they came up with a protocol for taking derived hepatocytes, individual endothelial cells, and mesenchymal stem cells, and they put them onto just directly onto matrigel. Okay, so matrigel, for if you don't know, is a basement membrane matrix that's primarily collagen 4 and laminin, and then it has a whole bunch of other stuff in it. So it's poorly defined. It's isolated out of a mouse sarcoma tumor. It's an extremely useful material for like angiogenesis assays and stuff like that. And at least what they showed was that over a relatively short period of time, that these cells would self-assemble and they'd aggregate back together and they'd form these cool tissue organoids that had blood vessels or vessel-like structures throughout it. We based what we were doing off of their protocol, and we had some luck. I mean, we basically got the tissue structure to form, but we really could never get vessel-like structures to form. I still think that using the adipose stromal vascular cells is a good way to do it. I think really the big issue is it's probably in part a question of structure density, meaning that when you put these things in, I mean there's no structure at all, and then as they start to assemble, it starts to collapse, it starts to contract, and these structures really become quite dense, and your formation of blood vessels, for the blood vessels to form and then start to invade and grow throughout, again, these 3D structures, it becomes more and more difficult the more dense your matrix is. One of the things I've thought about is, well, can you form it, and then can you then form a vasculature around it, and then get it to invade? We just haven't done that, but that's another way to do it, potentially. Other questions? Yeah.
So what was your experience with off-target effects on Cas9?
Yeah, so we did not find any, at least in our cells. Now we didn't, we didn't sequence the whole genome, okay? But the software that we use for designing the guides gives potential off-targets that to look at, to examine. We looked at, I think we published 3 or 4 that were in exons and 3 or 4 that were in introns, and at least for what was suggested by the software, we didn't see any. Now, again, that's one of the reasons why we also use the nickase. Because if you cleave the repair mechanism that the cell uses, and off the top of my head I forget what it's called, but the repair mechanism that if you cleave both strands of DNA, it's very efficient for repair, but it also is prone to introducing mutations. Okay. But the the homology-directed repair, which is what the nickase utilizes for repair, it's not as efficient, but it's much more precise. And that was one of the reasons, or one of the logic or rationale for using that specifically, is to also again reduce your potential off targets, because you're using this nickase and that specific kind of repair. Anything else?
All right, let's thank our
Okay,
speaker.
thanks.